So good afternoon, everyone. So welcome to the 2022 Christmas lectures, which is a traditional appointment that that ends sort of marks the end of the semester and the beginning of the upcoming holidays. Traditionally, this is held by ECLT. This is the center that I'm the director. I'm directing, but this year is the first event that we're running joint together with NICH, which is the International Center on Environmental Humanities that is directed by Francesca Tarocco, who is sitting here. Thanks to Roberta Raffaetta, who's made this possible. So for those of you who don't know ECLT stands for the European Center for Living Technologies, and it's a center that's is a network of 17, soon to become 18, because we're getting a new partner soon. International institutions. This was established within a framework of a European project in 2004, so it's nearly 20 years ago. And Irene Poli was the first director of ECLT. So this is quite an achievement. We're nearly 20 years old in this respect. So at that time, it was a project on artificial lives and living technologies. That was the core of the research center at that time. But now it's a modern research center. It's a hub for complex system at the wide, within the wide spectrum. And now we have this idea of partnership with the humanities to bridge the gap between these two usually disconnected fields of culture. So I just said that this is a joint event with the NICH. So please, Francesca, I believe.
Yeah. Thank you, Achille. Hi, everyone, and thank you to our speakers and everyone here and online. I'm Francesca Tarocco. I'm a historian of China, and I have the pleasure and delight to be running this new center that we have established at Ca' Foscari. It's called, we like this moniker, it's not an acronym, niche, as I guess the ecological niche. We are not trying to be particularly niche, we want to be big and broad. And we are partly funded externally and partly by the university itself. We have an advisory board, an internal advisory board of people from all departments of Ca' Foscari, including the sciences, of course, and the humanities. And we also have an international advisory board. So we work under the umbrella of the environmental humanities, but also trying to, in fact, reformulate some of the instances that have characterized the environmental humanities so far. And as Professor Giacometti said a moment ago, one of the things that we are trying to do indeed is to be truly interdisciplinary and transdisciplinary as well. We want to make the most of being here now in Venice, which is both a sort of a lab for environmental and climate crisis, but well, rather than it's a lab, we want to perhaps transform it into a lab for dreaming and thinking about change. So today, I think, is an interesting moment to celebrate, I think, the generosity and the open-mindedness of scientists like Margaret McFall-Ngai, who have indeed, before us, tried to follow that path. And so without further ado, the floor is yours. Thank you so much.
Roberta will introduce the speaker properly, please.
So, welcome everybody, and thanks for being here this afternoon. We are really excited to have Margaret McFall-Ngai with us, and thank you so much, Margaret, for being here. So, I'm briefly introduce her to you. I mean, will be long, but be assured that has been a selection, So, Margaret McFall-Ngai is the inaugural director for the Carnegie Institution for Science Cross-Disciplinary Research Division, the division of Biosphere Sciences and Engineer, focused on integrating diverse fields of biology from molecular biology to ecology. to tackle pressing issues such as climate change and sustainability. During the development of the new center in Pasadena, California, McFall-Ngai's lab is located at the Caltech, California Institute of Technology, where she is also a faculty associate. Prior to her current position, Dr. Margaret McFall-Ngai was a professor at the University of Hawaii, Manoa, and served as the director of the Pacific Biosciences Research Center from 2015 to 2020. She instituted the CMIC Center for Microbiome Research and was PI of the Keck Foundation Environmental Microbiome Observatory and an NIH COBRA grant, did I pronounce well, to investigate the role of environmental microbiomes on human health. She has also held tenured positions at the University of Southern California from 89 to 96 and University of Wisconsin-Madison, 2004-2015. Her research laboratory studies two areas. One, the role of beneficial bacteria in health using the squid vibrio model. And two, the biochemical and molecular design of tissues that interact with light. In addition, for the last 25 years, she has been heavily involved in promoting microbiology as the cornerstone of the field of biology, for which she worked in the White House during the Obama administration on the National Microbiome Initiative. She also co-organizes and runs an advanced summer course on microbiome symbiosis, on microbial symbiosis as a visiting professor at the Gulbenkian Institute in Lisbon. She was a Moore Scholar at Caltech, a Guggenheim Fellow, and she currently is a member of the American Academy of Microbiology, of the American Academy of Arts and Science, of the National Academy of Science, and an advisory committee chair for the Humans and the Microbiome program of CFAR, which is the Canadian Institute for Advanced Studies. She has also received a Doctor Honoris Causa at the EPFL, École Polytechnique Fédérale de Lausanne, in 2015. In 2017, she became Howard Hughes Medical Institute professor and the Advancing Science in American Foundation Scientist of the Year. So that would be enough, but it's not, because beyond her many achievements in the life sciences, she also is an inspiration for many in the social sciences. In 2014, she has been one of the keynote speakers together with people like Hannah Singh, Donna Haraway, Jenny Rardon, Ursula Le Guin, and so on, at the Anthropocene Conference, Arts of Living on a Damaged Planet, organized by AURA, which is the Aarhus University Research on the Anthropocene. So a very complete scholar, a role model for all of us, I think. So I'll let you the floor, and thank you again for being here.
Okay. So I think I'm going to stand up, if that's okay. I can stand up. I've never given a lecture sitting down, so I don't know if I can do that. But first of all, I'd like to thank Roberta and Francesca and Achille for the wonderful introduction. And this is such an honor and an opportunity for me to be here. I've learned a little bit about the fabulous frontiers that you guys are opening, and I'm very excited to be here and excited to learn about what you're doing. And now I'm exhausted because I'm like, oh, such an introduction. So I really appreciate it. What I'd like to do today is I'd like to talk to you about something I'm very passionate about. And that is that, oh yeah, thanks. I'm really passionate about the field of biology and where it's been and where it's going. And so what I'm going to be talking about is the fact that we are now aware of something that we couldn't have known is only about 15 years ago that we begin to learn about this world that we didn't know about before. So this is bringing about a revolution in this field. And I'm going to talk about that revolution, where it came from and so on, and then talk about why I think it's important now. So biology as a field, can we? Oh, yeah, just take me that whole, if it's possible to take that off. Oh, perfect. Okay. Oh, yeah. Okay. So biology, there's a saying in English called a sea change, which means it's a very big change in what's happening. So there's a sea change in biology, and it's driven by a better understanding that we have very, very recently of life's diversity and how the diversity expresses itself. The basis of the evolution of this revolution and why it is critical. So I'm going to put up this, I hope Kelly will appreciate that. I'm going to put up this piece and ask you to read it. Yeah, okay. Yeah, the bicycle. I love the bicycle. So this is, I was a philosophy minor as an undergraduate, and I love philosophy. And so I read this book about physics and philosophy by Werner Heisenberg. And Paul Davis wrote the foreword on this. And this is something that happened in the field of physics sort of in the 20th century. And the golden age of physics in the 30s and 40s. And what happened was, as the result of the development of this particular age of physics, the physicist's worldview was radical and irreversibly altered. And this was an amazing thing that happened. So I'm going to try to convince you that this is something that's happening in biology right now. So what have been the revolutions in biology in the past? So here I'm showing the revolutions in biology and when at the times in history, recent history, in which the worldview has been radically and irreversibly altered. So the first one I'm going to mention is Charles Darwin's theory of evolutionary biology, and the modern synthesis, which by Gregor Mendel, which provided a molecular or a mechanism for which adaptive change would happen. So this was a really big sea change in biology, and it was a very exciting time. And of course, it took a while for it to really take hold, because revolutions take a while. But it was really exciting. And the second one was the mid-20th century. So we got in the 19th century, now we're going to the 20th century. And this is the Watson and Crick, DNA as the genetic material, and with Rosalind Franklin, who provided the X-ray crystallography for what they put forward. So the mid-20th century, showing that DNA is the genetic material. And in both cases, this was a marriage of theory and mechanism. And so it's a very cool set of revolutions that happened in the 19th century and in the 20th century. So what is the 21st century revolution in biology, and how did we get here? And actually, it's all about a technological breakthrough. And I'm in Venice. So, so Copernicus had this idea that we were not a geocentric the planets and our solar system was not geocentric, but heliocentric. So he goes along, he has no real way to prove that. Well, this wonderful Venetian, Venetian from Padua, Galileo Galilei, comes down to Moreno and gets a couple of lenses and puts together a telescope that can actually show this was a technological breakthrough that allowed astronomy to be for certain that this is a heliocentric rather than a geocentric world. Okay, so this was the technological breakthrough in astronomy. So this invention, this major technological breakthrough of Galileo, changed our view of the universe, and it changed our view of our position in the universe. So this was a huge thing. So what's biology's current revolution? Well, for over 2,000 years, up until, well, even, so how many of you are biologists? Okay. How many, well, I'll ask you in a minute another question. So for over 2,000 years, biologists organized the biosphere optically. In other words, what we could see either with the unaided eye, and this is what we've got here with Aristotle, the unaided eye, Aristotle said they're animals and plants. And so what happened then was that Van Leeuwenhoek in the 17th century put together this small microscope up in the Netherlands, and he said that there are animals and plants. And then what he had done was he'd taken a little cheek swab from the inside of his cheek, and he put it on his little microscope, and he could see what he called animalcules. He could see microbes in there. He's the first person to visualize microbes. And so the next thing that happened was, these microscopes got better and better and better. And then eventually we have the electron microscope. And Thomas Whittaker, based on the electron microscope, back around in the 1970s, developed what is called the 5 kingdom model. Actually, in 1969 is when he first put it forth. But in this 5 kingdom model, he had animals, plants, and fungi, which are all multicellular or can be multicellular things. And then what he called the protists, which were microorganisms that had nuclei, and then the bacteria, microorganisms that did not have nucleus. Okay, and the thinking was that over evolutionary history, so the first things that happened, like, back in 3 to 4 billion years ago, was all prokaryotic or bacterial. And so it was thought that with each step, the next group of organisms supplanted the previous in diversity. And so that it was the thinking in the late 20th century was that animals, plants, and fungi were the most speciose of all organisms. Now, what's interesting about that is that how many of you have had the 5 kingdom model of Thomas Whittaker when you took biology? Yeah, how many of you learned that? Okay, it's really interesting for me because I go, even if I'm speaking to a group of first year graduate students in microbiology departments, we'll ask that question. It's very common that 50% of them still say that that's what they learned. And so this is the 5 kingdom model. And so what I say is from 1969 to 2008, I'm going to talk to you about why 2008 is important and beyond, because they're still teaching this, which is terrible. But the thing is that it used to be thought, for instance, that the number of bacterial species that were out there was about 7,000. 7,000 species of bacteria. That's the same number of species as there are species of snake. So very few. That's not a big number. Okay. So this is all based on optics. Okay. Now, What happened was in the 1970s, a guy named Carl Woese came onto the scene. He was at University of Illinois, Champaign-Urbana. And his idea was that to get an accurate picture, biologists needed a new method, one that really, really reflects the relatedness. In other words, their genes. Okay. So what he reasoned was that if you used a gene that is fairly well conserved, you can compare that gene across a broad swath of life forms and be able to discern the relatedness of those life forms. So this was done by DNA sequencing. What was really interesting about Carl Woese was at the time, in the early 1980s, I was a graduate student. And Carl Woese I was at UCLA, and Carl Woese came to speak to the university. So it was really interesting because the building that I was in had 5 floors. There were 4 floors that were the bottom floors of the UCLA biology group. Those bottom 4 floors were people who studied everywhere from molecular biology all the way up to ecology of plants and animals. And on the 5th floor, there were 9 microbiologists who studied microbes. There was not a single graduate student in the bottom 4 floors who could tell you the name of one of the faculty, or one of the professors on the 5th. There was no integration whatsoever. However, Carl Woese, who was a microbiologist biophysicist, came to UCLA to speak. And our professors in the bottom 4 floors, I'm actually an animal physiologist biochemist. The bottom 4 floors, the professors told us, you have to go see this guy speak. He is phenomenal. And so we went in and sat there when Carl Woese came to speak. And I remember thinking to myself, this changes everything. This changes everything. And that's when I started to realize how profound this was. The problem with Carl Woese at the time, or the problem that he had, was that sequencing was super slow and super expensive. And so this was, you know, there was nothing democratizing about this. He was a very rich guy, scientifically. So what happened, just like for Galileo inventing the telescope, what we have here that enabled the field dramatically was a technological breakthrough, something called next-gen sequencing, which happened between 2006 and 2008. But certainly by about 2008, it was very democratized. And so the cost of sequencing plummeted, and everybody and their mother went out and collected water and soil and took microbiomes from animals, plants, and fungi, and began to characterize the world in a different way. And so you can see, this is 30 years after Woese in 2008, and then this has only been democratized for 14 years. So it's like we've stepped onto a fast-moving train. In other words, it's just the knowledge is so, it's just exploding. So what have we learned? What have we learned so far in these 14 years? It's pretty remarkable. First of all, the biggest change in our view of the biosphere since Darwin, hands down. We know that there are not 7,000 species of bacteria. There are millions of species of bacteria and archaea. It turns out that instead of there being 5 kingdoms, all of the eukaryota, which we sort of nest into this group called the archaea. And so there is just a small branch within the archaea. So there are, instead of 5 kingdoms, there are now 2 domains, the bacteria and the archaea. So it's a huge, huge change. One thing that's really funny was that I was teaching at the University of Hawaii in 2017 or 18. And a guy named John Archibald from Canada was giving a meeting in Southern England. and he invited me to this meeting, and it was on symbiosis and on this sort of thing. Anyway, so John invites me to this meeting. And before I go to this meeting, Carl Woese had said that there were three domains. There was bacteria, archaea, and eukarya. In other words, big things. He said there were 3 domains. That's what Carl Woese said, there were 3 domains. So I go to this meeting. I teach this at Caltech. I'm teaching this course. Not at Caltech, Hawaii, sorry. And I say to the students, there are 3 domains. I get in the plane and I go to this meeting the next week, and a bunch of junior people have shown, no, excuse me, there are 2 domains. I get in the plane, I go back and I say, okay, what I told you was all wrong. Last week, what I told you was wrong. You know, this week, it's now we know there are 2 domains. And so this is turning out to be very accurate. So very exciting. And the eukaryota, what used to be these big crown groups, are now known just to be buried within the archaea. So the diversity of life is now known to be overwhelmingly microbial. There's absolutely no question about this. This was a world we couldn't possibly know. The reason why it had been the 5 kingdom model like this is because optically, bacteria have, excuse me, Ned's a bacteria, my partner Ned back there is a bacteriologist, and bacteria have so few features, you know. So what's interesting is that the bacteria, now we know there are, you know, all these species and all these different domains of bacteria. I mean, it's just huge. Animals and plants, sequencing didn't change our view typically. So there are thought to be between 30 and 40 animal phyla before, and they're still now known today, 30 to 40 animal phyla. And it has to do with the fact that that what you see very well reflects your genetics. With bacteria, they just are featureless. They're round, or they're oblong, or they're spiral. You know, some very, very small differences in the way they look, but they're vastly different in their genetics. So microbes are highly abundant. That's the first thing, and that they're everywhere. I mean, they're absolutely everywhere. In fact, you know, way up in the trophosphere, you find microbes. So what I'm showing here is their abundance. And so the thing was, not only were they featureless, but most of them you cannot culture in the lab. You can only culture about 0.1% in the lab, if that. And so what happened was, because, you know, this was a world you couldn't know, what happened was when they started to sequence them, they went, oh my God, you know, now that we can actually find out who they are, we couldn't know who they were. And if they were different, because they're not culturable. But you can query their DNA and ask, who are they related to, and are they really different or not? And so the other thing that they could do is they could show their abundance. So in seawater, you have about 10 to the 12th bacteria per meter cubed, or you have about 1 million per mil, about 1 million per mil. I mean, that's a lot of bacteria, yeah, in seawater. And so remember that when you're swimming on the ocean, now the terrestrial, when organisms made the water to land transition, it's very different. So when the ground that you're walking on has a lot of bacteria. So the soil has 10 to the 16th bacteria per meter cubed. Whereas air doesn't support, unlike water, doesn't support huge amounts of bacteria. So you can see by the numbers there that in the lower atmosphere and in the upper atmosphere, you have a lot fewer bacteria per meter cubed. And so there's a difference in the way that animals and plants and their evolution and their adaptation occur in water versus air, based on how many microbes are around. So I love this quote because at Carnegie, I work with astronomers and I work with Earth and planetary laboratory people. And I always say that you guys, the astronomers estimate that there are 10 to the 24th stars in the sky, but biologists estimate that there are 10 to the 28th microbes on Earth. And so it's yeah, close enough. Yeah, it's really a microbe-rich world that we live in. So what this means is, just like our view of our position in the universe, our view of the universe and our position in the universe changed with Galileo, Carl Woese, and then next-gen sequencing. This invention changed our view of the biosphere and changed our view of our position in the biosphere. And this is a big, big sea change in our thinking. So what we know now is that the activities of, and you know, it's only been 14 years, you guys. I mean, that's a really short amount of time. And so the activities of microbes are turning out to drive the form and function of the biosphere, and are absolutely essential and pivotal for the health of all sectors of the biosphere. Everywhere from plant health. So one of the things that you know, that we have to understand is that plants and their rhizosphere, you know, around their roots and so on and so forth, they have absolutely essential microbes that keep them healthy. And so, and the same thing is true for humans. As we all know, our gut bacteria, we now know our gut bacteria keep us healthy. And that's true, likely to be true of most, if not all, animals. Every time somebody has taken an animal and made it, you know, put it in a sterile environment or raised it in a sterile environment, a whole bunch of things are very, very different, and usually its fitness is dramatically impacted. Even if it doesn't maintain a persistent interaction or symbiosis, the presence of the microbial world has been something that shaped the evolution of animals and plants. So, and like I say here, it continues to shape the field of biology. In other words, we have to rethink so many of our premises based on this new knowledge. So let me give you a few examples of how things are like taking off like a rocket. So one of the things I love to do is I love to go to Europe PMC or to PubMed, which is sort of similar in the US. Europe PMC has a much broader coverage than PubMed does. It gives you more references. So if you type in the microbiome, which it means all the microbes associated with that particular thing, and the word soil, the word plant, the word ecology, or the word brain, you can see that the total number of references that come up when you do that search. And the most amazing thing is in the percentage of those that you see in the last 3 years. So in other words, these fields are just going up like crazy. So if you do this on PubMed, you can actually, PubMed does this thing where they show you, they have graphs on, and they show you what's there. So very similar to what, there are fewer, like I said, they're not as expansive in PubMed. But you can see how, what I got here, these little arrows that are there, that's 2008. You know, when we started to really be able to sequence, and just all these things are taking off like a rocket. And the funniest thing is, I was at UC San Diego. I was asked if I would consider being the dean of biology at UC San Diego. So I went down there, and there was this big search committee for this dean. And so there were, you know, the provost and a bunch of people. For those of you who know about UC San Diego, it's near something called the Mesa, which has Salk Institute and Scripps. And so they had all these people on the search committee, and a bunch of neurobiologists. And so I'm talking along, and I had decided that if I went to UC San Diego because, you know, there were lots of reasons, I would begin to try to integrate macro and microbiology into a single continuum for the field of biology. And UC San Diego is a very good place to do that. So one of the guys, this was 2011, actually. So one of the guys sitting at the table was a neurobiologist, very, very, very famous guy, like, you know, Nobel kind of guy. Anyway, so he looked at me and he said, well, the microbiome has nothing to do with the brain. And I said to him, well, you know, I happen to be on sabbatical at Caltech, and Sarkis Mazmanian, who was a microbiome guy at Caltech, and we're doing a graduate seminar at Caltech, and one of the students just presented the very first paper showing that the microbiome shapes the development of the mouse brain. And so I said to this guy, I said, you know, you might just keep an open mind, because I think that's probably going to change. And there it is. Look at the brain, how that's changed since 2008. It's very exciting. So one that really, really is exciting to me is cancer and the microbiome in cancer, which I find really interesting. And so what I've got here is what both your PMC and PubMed. I mean, look at before 2008, there were only 34 references, and after 2008, there were almost 14,000. The funny thing was, when I was doing this, I was doing this in my hotel room here in Venice. And the funny thing was, every time when I was doing it too slowly, because every time I would go back to look and make sure I had the number right there before, it was like, excuse me, stop. You know, so this is like at 10 o'clock at night on Thursday. Anyway. So what I'm showing here is that what people have found is that every type of tumor seems to have its own microbiome. And what I'm showing here is I'm showing a paper, actually an image from a review that was done by Rob Knight, in which he showed all of the data that are now available on this particular thing. And all these numbers have to do with the number of the reference that he has in his reference section in this review. And it has to do with diagnosis, mechanism, program, and treatment. And each one of those references in those different colors refers to a different dimension of cancer in the microbiome. But it turns out, you know, this, you can see that it's fairly recent, 2021 and 2022. And the extent to which whether or not microbes cause cancer, they don't cause cancer, in other words, is this cause or effect, or what exactly is going on here, is not really known. But it's exciting because Rob himself, he's a professor at UC San Diego, and what he's been able to do, which is, I think is very cool, is he started a startup company, and what they're able to do is take a blood draw, and with that blood draw, he can tell you if you have cancer and what kind of cancer you have. Because the metabolome, the activities of the bacteria in the tumor release things in the bloodstream and can tell you whether you have cancer and what kind of cancer you have based on these sort of markers. It's very interesting and it's very, very exciting, and a field is taking off like a rocket. So what we're finding here is that this integration is that organisms, animals, plants, and fungi occur as what we call nested ecosystems. In other words, they're not individuals anymore. So those of you who know ecology, it would be very common for people to model things based on, you know, they'd look at a single animal, and they would look at it as a single animal and model it in that way. So ecology is making big changes in the way they view the biosphere and how they model the biosphere based on the fact that we now know that this is a sort of ecosystem, and this ecosystem exists within is embedded in a larger system. So there is a set of nested ecosystems that make up the world. So the healthy human is a nested ecosystem, as many of you know, and so it depends on who you're talking to. It's now more likely that there are the same number of human cells in your body than there are microbial cells. So you're at least 50-50 bacterial and animal cells. When you think about it, though, the microbes, the metabolic rate of your microbes is huge. The metabolic rate is thought that as much as 70% of the heat that you produce is due to the activity of the microbes. So they're incredibly important for your biology. And in fact, if there are, whereas when you say the animal cells are one genome, the bacterial cells, there are 1,000 or so different microbes associated with it. And the estimate is that there are unique, when you ask for unique genes, the microbial genes are about 2 million and the human genes are about 20. So you're only about 1% human. I always say, look at the person next to you. That person is only about 1% human. That's cool. Yeah, and what is, yeah, there's a guy that's part of the Canadian Institute for Advanced Research who has this whole thing about asking the question of what does it mean to be human? Yeah, it's like, yeah, this is Tobias Rees. Yeah, at a very profound level, you know, and who's running whom? So for those of you who are really interested in this, Elaine Chao just published a paper that talks about how the microbes in your gut are responsible for you responding, being a person who responds to exercise and likes to exercise. Is that right, Ned? Ned read the article, I didn't read it. HSIAO, Elaine Chao. Yeah, she's at UCLA. And so they are responsible for making your heat. They're responsible for lots and lots and lots of your behaviors. So the problem is we have, as biologists, a formidable challenge is presented. And that is, this is this thing about thinking, integrating this critical new knowledge into our conceptual framework of biology is very, very, this is turning out to be really difficult. And why is that? Why is integrating these ideas difficult? Well, one of the things that happened when Watson and Crick and Franklin put together the idea that DNA was the genetic material, the field of a certain portion of the field, they brought in a lot of physical chemists and whatnot, and a certain portion of the field pivoted strongly toward molecular biology and physical chemistry. And what happened was the other fields went into deep silos. And so that is to say, you were an ecologist, or you were a physiologist, or you were a molecular biologist, or a cell biologist, or an evolutionary biologist. I mean, lots of very deep silos. And so these deep silos are in the United States, at least, and I don't know if that's the case here in Italy, but the silos are reflected in the structures of departments. So for instance, almost any university, you will have departments that are, you know, like cell development, molecular cell development, and ecology. There'll be two completely separate departments of biology. I happen to be on the advisory committee for the biology department at Harvard. So I went back there and we were sitting around and they were telling us what their plans were. And the last thing I heard was that what they were going to do was open a major of stem cell biology. I'm like, excuse me, you're going to have a freshman come in and the first thing you're going to stem cell biology. You know, we are at a place now where we can do better. And so in any case, getting this unification of biology with macro and microbiology unifying, I mean, it's a pretty exciting thing. So the structures of departments, institutes, universities, and funding agencies in the United States all have this problem. They're all highly siloed. And so trying to get, you know, talking across is a very difficult thing. So I show here the hierarchy of life. And so, like I said, you could be an ecologist, an organismal biologist, cell biologist, molecular biologist. But what we can do now is we can begin to unify this. And my sense is that the junior people are very much more open to this than the senior people, but they're in power. So don't forget. But it's I mean, this is something that will be your job is to get this sort of integration going. So one of the things that I often point out is an example is that in the US, and I'm curious about whether or not this is the case here, a college student can receive an undergraduate degree in biology, Bachelor of Science degree in biology, with a single lecture in their freshman year in introductory biology on microbiology. Single lecture. It can't be anymore. It can't be anymore. That's something that we've got to fix. So on the flip side of that, the microbiologists are very resistant. You go to the American Society of Microbiology meetings, and it's 10 to 14,000 people. They're all microbiologists. And many microbiologists have never had any animal and plant biology. They majored in microbiology, and they don't know anything other than that. What's interesting is that what I'm showing here is something that has shaped the field of microbiology since the 19th century. And that is on the left-hand side, that's Robert Koch and the germ theory and Koch's postulates and so on and so forth, pathogenic microbiology. And then Sergei Vinogradsky and Meyerink and people like that, environmental microbes. So microbes either made you sick or made whatever they were associated with sick, and then environmental, or they were out there in the environment, in the water, in the soil, and whatnot, doing who knows what. You couldn't know who they were, but they're obviously doing something. So there was this very, very strong dichotomy. And to this day, you can go to a meeting like the American Society of Microbiology meetings, and it's completely divided like that. There's a whole set of people who go off and talk about pathogenic microbiology. And what happens if the microbes are intimately in nested ecosystems in animals and plants? There's no home for that in the world of microbiology for the most part. Yuri might say it's getting better. Yeah, it's a human-centric view of the world. I mean, that's a really good point. And in the Anthropocene, where we're having this tremendous impact, and we have this really anthropocentric view of the world. So why is this important at this time? Well, we all know that we're in a very, very strong global crisis with global climate change. So I'm just showing some different things here, different parts of the world, and how it's impacting different parts of the world. But obviously we have a climate crisis. And we have to push this revolution forward, in my opinion. And the reason is we cannot possibly approach climate change in an effective and efficient way unless we have a very good conceptual framework for the biological world. Otherwise, we're going to be running around in tight circles and not, So what it reminds me of is when I was a graduate student at UCLA and my study site was the Philippines. I would go to the Philippines and I would travel all over those islands. And the way I did that was I would pay my way by speaking at universities. And I was doing very, very basic biology on a fish. And it had symbiosis, but it was very, very basic biology. And I would always be challenged by them saying, why are you studying all this basic biology? I mean, this is like, why aren't you studying fisheries? Things like, you know, something practical. And I would always say to them, in 1951, the fisheries biologists who were studying this fish decided that they would figure out how to more effectively gather that fish, or fish that get those fishes. In Manila Bay, 96% of the catch was this fish family. They did such a good job that 20 years later, 4% of the catch was that fish family, and they were not ecologically replaced. And I would always say to my audience, you've got to know the basic biology, you've got to have a good idea of how things are put together before you can manage it. And now with the knowledge, the new knowledge we have about the microbial world, this is absolutely essential. We know that they're at the basis of health, of everything. And like I was talking to you about these silos, we have these huge problems. So what I'm showing here is I happen to be on this, even though I'm not a microbiologist by training, I hang out with microbiologists a lot. And not only that, I was the chair of the National American Society of Microbiology meetings for 9 years. And so I know these people, even though I'm not one of them, I know these people. And we produced a report called, from the American Academy of Microbiology, called Microbes and Climate Change. And I'm the only non-microbiologist on this committee, and I kept raising my hand saying, you know, there are animals and plants out there, and those microbes are associated, but it's very hard to think in that way. And so this report is almost entirely just about this. And it's a very, very tough thing. So then the National Academies has a meeting, and it has a report that comes out, and it's nothing about that. So, and there's a US, I was asked to review the US projection for what they want to do for the next 20 years for climate change. And animals and plants are all over that thing. It's something that came out of the government. Animals and plants are all over that thing. Microbes, there's not mentioned once, not once. And so this is like horrific. The guy who was the major organizer, one of the major organizers for the American Academy of Microbiology task force, were on the left. He went to the meeting at the National Academy. It was specifically on climate change. It was a 3-day meeting. And he said he sat there for 3 full days. And the word microbe was not mentioned once. Yeah, he was an observer, and so he couldn't say anything. But, it's truly amazing, and I think it's very destructive. So I think we have to have some goals. We have to change this. And you guys, you young people, this is going to be something I think that I'm hoping that you guys begin to do this, and that is to integrate macro and microbiology. Unifying biology under a single conceptual framework. And this is going to be a big thing to do, but I think that you guys are open to it and have the capacity to begin to do this, and I'm very excited. And, you know, as such, you know, we can begin to do what chemistry and physics did in the 1930s and 40s, and that is to come up with a set of foundational principles that all biologists need to know. And that is beginning where the starting point is the microbial world, and then what are the nuances that upon that existing framework. To give you guys an example, those of you who are a biologist, what is the terminal electron acceptor? Sometimes, yes, thank you. Well, you're a symbiosis person. So the fact of the matter is, if you ask most biologists, what is the terminal electron acceptor? They will say oxygen. And, you know, microbes, if you want to know, that's the question. Given the physical background of what the Earth presents, what are the possible terminal electron acceptors? And you will, the microbial world has how many, Ned? 15? Something like 15 terminal electron acceptors. So you get an idea of what's possible for life forms on Earth. And then you say, when a eukaryote comes around, big bang for your buck. It's the biggest bang for your buck is oxygen as a terminal electron acceptor. And that's why the radiation of the eukaryote was so successful. But the fact of the matter is it's not the foundational. You know, you want to go back to starting each topic with the microbial world and building out from there. But I wanted to leave on an up note. I didn't want to be all negative here with everything that I was saying. And so what I have here, change is happening. And so in reforestation events, people are beginning to recognize that in reforestation, you need to include for those of you who are biologists, the mycorrhizae, the fungi that are associated with the roots, as well as the bacteria and the archaea that are in the rhizosphere. They co-evolved with those plants. So how many of you guys know Larry Ellison? Does that ring a bell? He's one of those big .com guys. What did he? Can't remember. You know, one of those .com things from Silicon, hugely rich guy. So Larry Ellison buys the Hawaiian island of Lanai. He buys the whole island, okay? And at one point, I was at the University of Hawaii at the time, and at one point he decided that he would reforest Lanai with natural native Hawaiian plants because it had been a pineapple plantation for decades. It had been just one pineapple plantation. We decided to reforest it. He couldn't get it to work. He would go off and he'd bring it, you know, with no mycorrhizae, put it in the soil, it died. And he figured out, he kept doing this and it didn't work. So he invited the Simaiki junior people over from the University of Hawaii to talk to him about why it wasn't working. So he brought the people who studied fungi and bacteria and whatnot associated with the soil. And maybe at some point he'll get that to work. The other thing that I'm showing here are coral reefs. And so we're beginning to genetically engineer the zooxanthellae and the corals themselves to be more tolerant to climate change. And so there's a guy, Phil Clevis, in my group, the Carnegie, who's doing this very thing at Johns Hopkins. Also, in putting together this talk, I came up with two really cool articles that I thought you guys might be interested in. And so one of them is about ameliorating drought stress with the bacteria. So it turns out that you can do a lot by changing the microbiome of organisms. So, and then the very last one, this Lee et al. It's actually 2023, it says. Experimental temperature shape host microbiome diversity and composition. This is so cool. So what this group in Oxford, they're at Oxford, what they did was they characterized the microbiomes of 45 aquatic invertebrate and invertebrate and a terrestrial vertebrate and invertebrate animals. And they did a series of temperature experiments and showed how those microbiomes changed with temperature. And so that's a very cool article. Actually, I wrote to her because I couldn't connect to get the reprint, and I wrote to her and she sent it to me right away. It's really nice. So we have to do a couple of things. We have to change our conceptual framework, like I was saying. So here are some goals and strategies. So as I mentioned, we need to integrate macro and microbiology, and then we have to engage physical chemists, scientists, mathematicians, anthropologists, sociologists. And that's why in putting together this talk and looking over what you guys were doing, I'm so very excited, because you guys are really at the cutting edge. I happen to have the pleasure of being on the National Academy New Biology, the 21st Century Committee, where a dozen scientists from around the country, for 2.5 years, we met to tell the Congress where biology was going to go for the next 20 years. Well, we came down to the idea that the global problems that were looming have biology at their base: food, health, energy, and ecological environment. Those were the 4 ones that were identified. But many of the solutions are going to come from engineering and physics and math and chemistry and sociology and so on and so forth. So what you guys are doing is just really phenomenal in that way. And so this is something I put together for the website. But the leadership in integrating these processes is happening. And you guys are doing that big time. What we're doing at Carnegie Caltech is we're looking again at the rules of life and seeing how to integrate the whole darn thing, and then nested ecosystems, all with the vision for how do you make things sustainable and resilient in the face of climate change. I wanted to, now this is a bold advertisement. So those of you who are interested, before I finish, I wanted to mention this. So I teach every other summer at the Gulbenkian Institute in Portugal. And this is a European network of microbiome research, and CIMNET is, and it's just an amazing group. And the Gulbenkian Institute in Oeiras, Portugal, every 2 years has a 2-week summer course in symbiosis that is offered in July. And we're offering one this summer. Typically, as I show here, there are 30 to 35 students and about 20 faculty from around the world. And we come together in a very, very intense 2 weeks and sort of explore this topic. And the applications are open soon, so any of you who might be interested, it's a really, really fun time. So grazie mille.
Thank you. I'm sure that, thank you, very, very fascinating. You changed my perspective. Yeah. Ranking of discipline in biology has been always very high, but not for field. Now we have changed it. I've changed my mind. So any questions for Margaret? Sandro, please.
So thank you very much for this very nice talk. I just want to ask you, so in view of what you presented, what are the chances that we find some microbiome somewhere else in the solar system? And if yes, how much do you expect this microbiome to enlarge the phylogenetic tree that you presented, now that you have shown to us that our view was so reduced? So much will an extraterrestrial microbiome enlarge? Do you expect it similar, or do you think it will make it 10-fold bigger or a million-fold bigger?
Actually, we're still not to the asymptote. In other words, I don't know the answer to your question because we're not even approaching. So the thing is that Terra Oceans, we were talking about, they went across the Sargasso Sea and collected every mile or something and analyzed the microbes. And it's just the sequencing is becoming cheaper and cheaper and cheaper. So you can do this like at amazing speed too. Yeah. Yeah. So with the outer space thing, it's really interesting because you talk to people who are doing this sort of thing, and they're beginning to rethink all sorts of things. And one of the things is the extent to which abiotic processes organic materials, can synthesize organic materials. So I was just talking to one of the guys in Earth and Planetary Labs because I said, do you believe that? And he was the person who did the analysis, but they overrode. I think he's even on the paper. And so the thing is, there some need to question. The other one they question now is in the spot. At the that the banded iron formations were the result of abiotic activity. So there's all this confounding stuff. And, you know, as far as, you know, life on other planets, this is fantastic. So there's this guy at, so you're looking for other things on it. What they do is look for water, or they look for some kind of carbon compound or DNA or something like that. And they're looking for, you know. So this guy at EPL, he was at Caltech and he's at Earth and Planetary Labs in Washington, DC and Carnegie. And this guy, Michael Wong, is his name. So I happened to go to a board of trustees meeting where presented. And so what he's done is he characterized the networks of molecules in the atmospheres of all of the planets of our solar system. So there are networks, the atmospheric, that all of the planets from Mercury, Venus, and so on, all have identical networks, except Earth's networks. The networks are like neural networks and biochemical networks. They are basis of life. So it's fast because you wouldn't think Saturn, Mercury, I mean, for him, they do. Earth is really different. Yeah. Now they can go out in very deep space, and based on, as I understand it, they can look at the radiation of those planets, and they can say, in a given solar system, is there an anomalous planet? And is it possible that that anomalous planet is anomalous because it has something like life? So these are the kinds of things that I've been listening to at Earth and Planetary Labs. Yeah. Well, and you know, one of the most amazing things is they keep finding bacteria like in the deep subsurface. I mean, it's like there's one place where there's a single bacterium, and that bacterium, I can't remember the name of it, but it does everything. It does everything, but it's the only thing there. Yeah, and it has to do everything. It's the only thing there. But you know, I think we have, I guess, at the end of the day, the biggest message is that we haven't even scratched the surface yet. We haven't scratched the surface.
There's the question.
the next technological breakthrough? That's a very good question.